So Chapter 10 deals with gene action or gene expression. And what that means is we're going to take the information encoded within our DNA as nucleotides and we're going to first convert it into RNA, which also has a series of nucleotides, and ultimately translate that information into a string of amino acids, and those are the building blocks of our protein. So this is the central dogma of biology, the idea that we're taking the genetic information within our cells, stored as DNA, we're going to do a two-step process where we convert it to RNA and ultimately to proteins. And this is important because we've talked about how our genetic code, the series of bases that make up our genetic code, ultimately codes for proteins. Obviously, not all of our genome does this because we've learned it's only a small portion, or the exome, that are actually expressed. But to actually get at that information, we need to go through a series of two steps. And the two processes that we need for all of this to take place are transcription, which takes place within the nucleus. And our goal here is the first step of this pathway. So we're taking our genetic information on DNA. We're going to convert it into this little M, or messenger RNA transcript. And that's important because this transcript can actually leave the nucleus and go into the cytoplasm. So we want to maintain a copy of, at all times, of our DNA within the nucleus because our bodies need to make proteins constantly. So you don't want to lose that original information. And so this allows us to take a little transcript or a little copy of that information and ship it out into the cytoplasm so that the second process, which is translation, interpreting that string of codes as a series of amino acids, takes place in the cytoplasm. So this chapter I've broken down into two different lectures. Part 1 is going to focus on transcription, and part 2 is going to focus on translation. So the learning outcomes associated with this chapter, this first part we're going to cover the first bullet point. We are going to learn about the 3 major types of RNA molecules and what their functions are. We are also going to learn about the importance of transcription factors. So they're involved in the process of transcription. And we're going to walk through the steps in this first part of the lecture of transcription, and then in part 2, translation. We're going to discuss how researchers figured out the genetic code. So how this all works. How is it possible that we have just 4 bases, right? Our A's, T's, C's, and G's, but they code for all of the different proteins and phenotypes that we see in our body. So we're going to talk about how that works. We're also going to talk about 4 components of a protein shape. So once we have our protein at the end of translation, we have to go through a series of modifications of those proteins in order for the protein to finally achieve its ultimate function, and that's related to its form or shape. And finally, this kind of ties into the last bullet point. We're going to talk about protein folding and what happens when things go wrong. So I already mentioned this on the overview slide of the lecture, but everything that we're learning about within this chapter all falls under this idea called the central dogma of molecular biology. And this dogma was proposed by Watson and Crick in 1957. We've already talked about these two scientists. So they contributed to our understanding of the structure and composition of DNA, along with Rosalind Franklin. While they were working on their building of the structure, the double helix of the DNA, they also discovered that all of this information within DNA is ultimately turned into proteins. So it's the idea that we've got two different steps. Yeah, spelling error there. The directional flow of information. So we're going to transfer all the information stored within our genes in DNA, and we're going to convert that information ultimately into protein. And proteins are what create or influence our phenotypes. Because phenotypes are the physical expression of the underlying genes. So all of this information that we're going to be transferring during transcription and during translation is all contained within the specific sequence of nucleotides. Okay, so remember that nucleotides are the most basic unit or building blocks, the monomers of DNA. And nucleotides include a sugar group, and it's deoxyribose when we're dealing with DNA, phosphate group, and the key here is the nitrogenous base. So those are adenine, thymine, cytosine, and guanine, or A, T, C's, and G's. So it's this sequence, the actual sequence of the bases within the nucleotides that encode the information. And the way to think about this is, as I have on this page here, we have letters of the alphabet, and we use those letters to come together and create words and create sentences, create pages within a book. So it's all in the key of how we organize our letters. But while we're dealing with DNA, turning it into protein, it's interesting because instead of our 26-letter alphabet that we use in English, we have just 4 bases. So I've already mentioned to complete this task, we're going to go through 2 steps. First, we're going to take the DNA and we're going to transcribe it, right, or make a little note about it, if you will, into RNA. And specifically, a template strand of mRNA. We'll talk about that more in a minute. And then the second step of this process is taking that code stored within our RNA and translating it or interpreting it as a series of amino acids that build up or make up a protein. So when we look at the definitions of these 2 terms, it helps us to think about exactly what we're doing when we're dealing with gene expression. So to transcribe something is to make a written copy of. To make a copy, a dictated, a recorded manner in longhand or on a machine. So we are going to make a copy of the genetic information. And we're not going to copy the whole double helix, because if we did that, it'd be confusing. We'd have 2 sets of information, and it wouldn't be able to leave the nucleus. So instead, we're going to make just a single little strip or a copy of a single strand, which ultimately is a complement to the other, so that it can leave the nucleus. Step 2 is to translate, and this means to turn into one's own language, to transfer or turn to a symbols into another, express in different terms. So essentially we're expressing the code or the copy of the code. So we have a copy of a code, which in this case, our original code was DNA, and our copy of the code is our RNA, and we're going to express that differently. We're going to transfer it or convert it into something new, and in this case, it's going to be directly amino acids, and a string of amino acids together make up a protein. So if DNA is our code, our genetic code, what does it actually code for? So strictly speaking, DNA actually codes for RNA. And RNA, as you know, is just another type of nucleic acid. And it differs from DNA. This was one of the homework questions by the sugar that makes it up. So instead of deoxyribose, we have ribose. It's also single-stranded, where DNA was a double helix. And there is a base that is different between DNA and RNA. So we still have our adenosine, our cytosine, and our guanine, but instead of thymine, we have uracil. And there are different forms of RNA. So 3 different forms that we're going to be learning about. And I want you to keep track of what their functions are, so you're familiar with what their jobs are in terms of gene expression. So we have messenger RNA, or mRNA, just as it sounds. It's a messenger. It's going to relay the code or the information as an intermediate between our steps. We have a transfer RNA, or our tRNA, and the tRNA is involved in translation. So it transfers the specific amino acids that correspond to the codons or triplets of bases on the strand of RNA. So their job is to transfer things. And our ribosomal RNAs are RNA, and that makes up our ribosomes. And our ribosomes are the location where translation actually occurs. So it offers a place that supports or provides support for all of these steps within translation to take place. So there are some other RNAs, but I'm not going to go into depth on the rest of those. We're going to focus on these 3. So strictly speaking, DNA codes for RNA, but ultimately DNA codes for protein, right? That's the big part of the central dogma. And protein is going to be our end product. But we're not made up only of proteins. Just keep that in mind. So how does an organism know how or when to build or break down all of these different macromolecules that we see in our body? So our carbohydrates, our proteins, our lipids, our nucleic acids, all of the other things besides proteins that we're made up of. Well, that is due to things called enzymes. And enzymes help facilitate or speed up chemical reactions. But interestingly, they are also proteins. So this slide gives us a nice overview of 3 different processes that we've learned about. So DNA is our genetic material that's stored within the nucleus. So this is just a reminder. In Chapter 9, we covered how this genetic information, in order for it to be passed on to the next generation of cells, we need this process called replication. So we want to make identical copies of this information so that it could stick around for the long term. So that's replication. And that happens during the cell cycle, during the S phase of interphase, or during synthesis. That's the only time it happens, during the cell cycle. Now, the process of transcription, as we've just discussed, takes all of the information on DNA and turns it into RNA. And that happens also within the nucleus. And the process of translation is ultimately creating a protein made up of these amino acid building blocks, using the information we're going to pass via RNA out to the cytoplasm. So while I'm on this slide, I just want to remind you that in order for gene expression to happen, for our first step of transcription to start, we don't have to have replication come first. So I noticed that in reading over your textbook, it almost makes it seem like these 3 processes happen in this order. But that's not really the case. Replication, we know, takes place during interphase. Transcription and translation, because they are both parts of gene expression, are happening throughout interphase of the cell cycle. So during G1, G2, and synthesis, any of those times it can be happening. Whenever the signals start, as we'll talk about in a minute, the cascade of signals and steps that happen to start gene expression, whenever those start, it happens. And they can actually, transcription and translation, can actually be happening during the same time that replication is happening. Translation and transcription, however, cannot happen at the same time. And that's because in order for us to translate something, we need the transcript first. So just something I wanted to clear up. So gene expression relates directly to the transcription and translation. 2 processes where we're going to change that information contained within DNA, convert it into RNA, translate it into protein. So the portion of the human genome which actually encodes for protein or encodes the specific string of amino acids is called our exome. Okay, so the exome is just a small component of the larger overall genome. Okay, so it's a very small portion, and it's like 1.5% of the whole genome. That's it. So the very small proportion of the genome and all of the bases and chromosomes that we have, less than 2% of it actually relates to the production of proteins. So the rest of the genome actually helps control the process of protein synthesis or gene expression. So all of the other stretches of DNA, a lot of them, their functions are to help control the two processes involved in gene expression. So they help control transcription and translation. So those controls involve including the time in terms of when things start or stop, the speed at which things occur, and the location in terms of where things are happening within the body or which cells are actually undergoing protein synthesis. And our genes, so the exome itself, is encoding 20,325 different types of proteins. So even though the exome is only a small part of the overall genome, it actually encodes a lot of different proteins, all the different proteins that we need. And we're going to talk about how this is even possible in later slides. So gene expression, taking that information and turning it into the physical phenotype or the production of protein, and from these instructions involve several steps. I feel like I keep repeating this over and over, but I think it's important because it'll help you keep track of what's happening. So we've got transcription, process 1, and translation, process 2, are 2 steps of processes that we're going to go through. So transcription, we're going to synthesize RNA. Translation, we're going to use that information in RNA to manufacture a protein. And how we're ultimately doing that is by joining these little amino acids. Finally, we don't end there. In some cases we do, if the protein is really simple. But typically, we have to actually fold the protein, to go through a series of different steps to fold the protein into a very specific 3D form. So proteins all have very special forms, and the special forms go hand in hand with their specific functions. So we'll talk about how this works way later in the lecture. So this table gives a nice overview to show you that even though all of the proteins in our bodies are made up of the same 20 amino acids, so we keep that in mind, right? So we have, to me this is really fascinating, we have only 4 different nitrogenous bases, right? Are A, T, C, G. Uracil when we're dealing with RNA. And these 4 different bases, because we can put them in different combinations, a series of 3 letters called codons. There's 64 of them. Encode 20 different amino acids, but ultimately it's the arrangement of those amino acids that then produces this 20-some thousand plus proteins in our body, which is crazy. So here this gives a list of all of the protein diversity that we see in the human body. So on the left we have the different types of proteins that we see. Actin and myosin we see in our muscles, so muscle fibers, and it enables muscles to contract. Antibodies and antigens, those are all proteins involved in immunity. Collagen and elastin, things that we see in the connective tissues within our bodies. So within connective tissues proper that support the dermis of our skin, to things within our joints, our vertebral discs within our spine. Obviously, these enzymes, which we're going to learn a lot about in this lecture, anytime you see an ASE at the end of something, it means it's an enzyme. And all enzymes are proteins. So these enzymes are really important in DNA replication and gene expression. Hemoglobin, probably familiar with, so oxygen transport. Insulin, control of blood glucose. Keratin builds up our hair, our nails, our skin. So lots of different things within our body. So throughout both of these processes, we're going to be dealing with our two different macromolecules of nucleic acids. So there are just two different types of nucleic acids. DNA, so deoxyribonucleic acid, and RNA, ribonucleic acid. And although they are both made up of the same monomers, these nucleotides that at their most basic level have these nitrogen bases and then sugar phosphate backbones along the outside that anchor them, they do differ in several aspects. So this table is a great way to compare and contrast DNA and RNA. And the homework questions from chapter 1 actually covered a lot of these because it was included in the general overview of the textbook. So most students did a really great job on listing the comparisons and contrasts between these molecules, but we're often missing one of the key components. So this table is a nice summary of what's going on behind them. So DNA is usually double-stranded, so that double-stranded helix structure. And RNA is usually, although not always, single-stranded, kind of coiled. So we've talked about this. So we've got 3 of the same bases. So adenine, cytosine, and guanine are the same in both cases. The only thing that differs is the 4th base. So DNA has thymine, RNA has uracil. The sugars differ, the actual chemical structure of it. So DNA has deoxyribose, an oxygen unit within the sugar, and RNA, based on its name, has ribose. The functions of DNA and RNA are different. So kind of the last 3 pieces here relate to their overall function, whereas the first 3 relate to structure. So differences in structure, but because in biology, form usually, or always, leads to function or relates to function, we also have functional differences. So DNA maintains protein coding information, right? And that kind of goes with our last thing on our list here. It persists. So it's going to maintain that information within the nucleus of the cell across generations and over the lifetime of the cell itself. RNA, on the other hand, is going to carry the information. So its job is to kind of transport this information from place to place and also control how that information is used. DNA stores it in the long term. RNA carries, transfers it, interprets it, controls how it's used. DNA cannot function as an enzyme, but RNA can, which is really interesting. Okay, and as we've just learned, so RNA, because it's involved in transferring things and carrying things, it's transient. So it forms and then breaks down again, all in the course of these different steps that we're going to see. It never stays within or put together within the nucleus or cytoplasm. So this is figure 10.4 from your textbook, and this just shows visually what we just went over in terms of verbiage in the table on the previous slide. So we're covering the differences between DNA and RNA. So DNA stores RNA and protein encoding information, and then it can transfer that information onto daughter nuclei or cells. So this is the long-term persistent data storage. So in order remains within the nucleus, we can copy that information, make sure an identical copy is passed on to the next generations of cells via replication, and passing it on to the next generation of nuclei via mitosis, and also passing it on to the next generation of individuals via meiosis. So RNA carries or transfers the protein encoding information and also helps to make our proteins. So that's why this is transient in nature, because it's very short-lived and the job is to transfer information and help interpret it. So you can think of this as a little jump drive or memory stick we would use to transfer information and then see a final product. So then we have some structural differences. So here we have a double-stranded double helix of DNA versus our single strand of RNA. Our sugars differ. So we see we've got a pentose or 5-carbon sugar in both cases, but it's all about a functional group located at the 2' carbon. So to count our carbons, 1, 2. So for deoxyribose, we simply have a hydrogen group bonded to that carbon, whereas in ribose, we have a hydroxyl group. So that means we've got this oxygen present. So that's why DNA, because it is missing that oxygen, it's called deoxyribose. It also differs structurally with regards to the bases that we see. So 3 of them are the same, cytosine, adenine, guanine, but one is different. So these, remember, are our pyrimidines and GR purines. So one of our pyrimidines is different. So instead of thymine that we see in deoxyribose, we have a uracil. And you'll notice that structurally they look extremely similar. The only difference is in one of our groups. So thymine has this thing called a methyl group that we don't see over here. We just have a hydrogen unit. Here we've got 3 of them, so it creates a methyl. So interestingly, I was just looking up, like, why is there this difference? Why do we see a different base in DNA than we do in RNA? Well, the hypotheses are that RNA as a macromolecule came first. So this is the simpler of the 2 structural units. We only have 1 hydrogen, right? But in DNA, we actually have 3. So what I've read, the idea or hypotheses behind this is that structurally, thymine is less prone to breaking down and binding with other things, whereas uracil is easier to break down and actually can bind with a lot of other bases, strangely, and sometimes even itself. So this change, evolution, or appearance of this new base for information, genetic information, helps maintain that information, excuse me, because structurally it sticks around longer and is harder to degrade. So here's the overall structure of RNA in terms of looking at the building blocks or monomers of RNA and their smaller components. So just as we saw with DNA, our monomers are nucleotides. More specifically, they're actually called ribonucleotides, whereas in DNA they're called deoxyribonucleotides, but you just need to know they're nucleotides. And so because they're nucleotides, they're made up of the same three components. So we have our sugar, but in this case, remember, it's ribose because we have this hydroxyl group at the 2' sugar. We have our phosphate group, which looks exactly the same. So this is our backbone, our sugar phosphate backbone. And we have our nitrogenous base sticking out into the center that forms bonds between one another. And we've just discussed this. So our purines are adenine and guanine, which is the same. For pyrimidines, we also have cytosine, but we see uracil instead of thymine. It's usually single-stranded. It still has 3' and 5' ends, right? In this case, this is our 3' end because that's where the sugar end. This carbon is our 3' sugar, and it's sticking off that end. And the other big key is that it's much less stable than DNA. And that's one because, as I just mentioned on the previous slide, we've got uracil instead of thymine, but it's also because we're dealing almost always with just a single strand and not a double helix. So it's less stable, it's more transient in nature. Okay, so now let's get into the nitty-gritty of this. So remember, this lecture is going to focus on the first of our 2 steps of gene expression or protein synthesis, and that is transcription. So the overall goal of transcription is to take the information encoded within DNA and we're going to turn that into RNA, right? That's step 1. So our RNA really is what forms the bridge or the connection between our genes within our DNA and our end product, which is going to be protein, since we're dealing with protein synthesis. So you can see how this is the connection between these 2 pieces. So more specifically, we're going to take the information on DNA and turn it into a transcript of M, or messenger RNA. And the bases along that mRNA transcript or sequence are complementary to those on one strand of our double helix. So remember, we're going from a molecule that has 2 strands to a molecule that only has 1. So to do that, we're going to open up our molecule and access the information, and we're going to make a little transcript or copy. So there are 2 pieces of our DNA helix here. So as you can see in the bottom, we have this all unzipped because we have 2 strands that make this up. One strand we call our coding strand. So that is the actual information that we're going to send out of the nucleus. The only difference is going to be every time we have a T, we're going to have a uracil instead of a T. But you'll notice that this mRNA transcript down here is exactly the same nucleotide sequence or base sequence as we see in our DNA coding strand, the exception of U's replacing T's, okay? And we can do that because when we build this strand of mRNA, we're going to use the other half of the double helix as our template. And that's why we say that this mRNA transcript is complementary to our template strand, because these bases can form bonds with one another, hydrogen bonds. So A's always pairing with U instead of thymine, G's and C's as we've seen before, okay? So RNA polymerase is the enzyme, the major enzyme that we're going to see, whose function is to build this mRNA molecule or mRNA transcript. So here at the bottom of the slide, this is just showing you an image of where this is actually happening, since this is an overview of transcription. So just a reminder, we're still hanging out inside the nucleus of the cell. So that was step one, transcription, right? So our DNA to RNA, right? That's our bridge. And translation involves taking that RNA and converting it ultimately to protein. So that's the goal of translation, taking the information encoded within the instructions and turning it into a protein. So that means interpreting it as the series of amino acids that are encoded within the DNA and creating a protein. And how this works, so this takes place, as I've talked about, in the cytoplasm, the cell. And how this works, we'll learn that a series of 3 adjacent, 2, 3, nucleotides on our strand or template mRNA is called a codon. So now we still have the same information that we saw within our DNA helix. Remember, from our DNA coding strand, right? Our DNA coding strand and our mRNA template look exactly the same, except we've got uracil instead of thymine. Here we've got, in this image, our DNA template strand, right? So to make our mRNA, it's complementary to what we saw on the template strand. And now the key to this all is that even though we have the same series of bases, except for our uracils, as our DNA coding strand, we can use that information by looking at series of 3 bases at a time. And that is the key. Instead of translating each of these bases one by one, we're going to see that we translate it based on the codons. So each codon or series of 3 bases encodes or corresponds to a specific amino acid. So you can see how all of that information, even though it's just a series of bases, we read it as a series of 3. We have different amino acids that can then bond to one another to make a long string of amino acids or a polypeptide. And ultimately, we can fold a string and do other things that we'll talk about later to create our final protein. So I'm going to jump ahead a little bit. The way my lectures organized, the way I like to organize these processes is kind of giving a broad overview of each of them and then talk about what the genetic code is before I go into details. And your textbook just goes into the details first before discussing the code. So I'm going to jump ahead to what our genetic code really is, since that's what we've been discussing this whole time. So we know at this point we have 4 different bases, and we know that there are 20 total amino acids that our body needs for proper form and functioning. So when thinking about how this code actually worked, or this bridge between the genetic code and proteins, so scientists had to lay down or think about certain requirements or how this code would really work. So they knew we have 4 total bases and there are 20 different amino acids. So in order to figure out how this code works, they thought about, well, how would these bases kind of come together in order to make 20 different amino acids? So there's no way that each base would stand for just one amino acid, because then there would just be 4 of them. If 2 bases encoded an amino acid, that still wouldn't work, because we've got 4 as our base unit, and we raise it to the level of n to give us how many different possibilities we would end up with. So 4 raised to the 2nd power would give us only 16 amino acids, and we know we need 20. So because of this, they realized that those bases need to be read in a series of 3 in order for us to cover all of the amino acids. So because we've got series of 3, it actually gives us 64 different combinations. So we could potentially encode 64 different amino acids, but there aren't that many. There are only 20. So the cool part about that is you'll see, looking at this chart, that many amino acids actually have multiple codons related to them. So the multiple codons actually encode the same amino acid, and we'll talk about why that's important in a second. But this first piece, we know our DNA code, or our genetic code, is based on triplets. And these triplets are called codons. Okay, so here we've got all of our codons, the different potential combinations, and we read this genetic code based on here. Over on this left vertical side, you start with your first base, and we're dealing with, remember, RNA at this point, because we've got our RNA transcript and we're going to translate it into our proteins or amino acids. So instead of T's, we have U's. So you look up your first base, and then your second base runs horizontally, and the third is over here vertically on the right. So it's just a nice way to organize all of these 64 different potential combinations that we see. So 61 of these codons actually encode a specific amino acid, but there are 3 of them over here that actually don't encode an amino acid, but they are going to send the signal for translation to stop. So we call those our 3 stop codons. So here's our universal genetic code. So on quizzes or exams, when I'm asking you to translate an mRNA transcript, you need to look up the codons on this chart. So some other interesting things to note about our universal genetic code. This code is unambiguous. And what that means is that each codon only codes for a single amino acid, right? So we see a codon, and we know unambiguously, without a doubt, this codon only codes for the amino acid methionine, right? So it's unique in terms of the fact that they're going to line up with exactly one and only one amino acid. The next key is that our genetic code is also redundant. So we've just discussed that because codons are a series of 3 bases, we end up with 64 different potential combinations, but we only need 20 amino acids. So what that means is that most amino acids actually have more than one codon that codes for them. And we saw that in the previous chart. So sometimes there are some amino acids that can have 4 different codons that code for them. So even though it's unambiguous in that our codon is only going to match up with or signal for a specific amino acid, that same amino acid could have, and I'm making this up right now because methionine is that, but I'm just pretending. I'm giving you an example. You could have multiple codons encode the same amino acid. So we refer to that as the redundancy of the code. And another term for that that's used in your textbook is degenerate. Okay, because we have so many possibilities, most codons have more than one, most amino acids have more than one codon that can specify them. And that's important. We refer to this as the wobble effect. And we'll talk more about this when we get into mutations. But the really great part about that is that even if we have a mistake in the genetic code, oftentimes, if it's just a mistake, especially in the 3rd base, these codons will typically code for the same amino acid. And the cool part about that is even though we have a mutation, it actually doesn't have an effect because of this wobble effect. We still have the same amino acid that we had in the first place. As I mentioned, we will go into way more detail on that when we cover mutations. But it's something to keep in mind. So unambiguous means what each codon encodes a specific amino acid. The redundancy or degenerate part means that because we have so many potential possibilities, we can have more than one codon code for the same amino acid. So we also have some controls built into our code. We have a start codon and we also have multiple stop codons. And this is important because we've got this long string of nucleotides and our machinery needs to know where to actually start reading this code, to start associating amino acids. So our start codon is our AUG codon. And it actually encodes this methionine amino acid. But it signals for all of the machinery to actually start. So our ribosomes are the site in which this is going to actually happen. That's where it starts. And then we've got more codons after that. Until we reach one of our 3 stop codons, which do not encode an amino acid but signal for the stop. So we've got some controls built in. And these controls relate to our reading frame. And I'm not going to talk about that as much right now because we'll go into it more when we cover mutations. But our reading frame is simply the order that we read, or the ribosome reads a sequence. So it's just the series of codons and then how they respond to amino acids. Because we'll learn that if there are certain types of mutations, can alter our reading frame. It can alter the way the sequence is ultimately read by the ribosomes, which then leads to different amino acids and a different protein being produced. So the final piece to this really applies to this universal part of the theme here. Every living organism uses this code, which is pretty awesome. So it's the same in all species. So all species have the same mRNA codons for the same amino acids, all of those 20 amino acids. So we have the same genetic code. And the simplest explanation for this is that evolutionarily we all were derived from a common ancestor. And this code, this sequence, and the way that it corresponds to amino acids is extremely efficient in the way that it works. It can be passed on, it persists over time, and therefore was likely passed on to descendants. So the cool part about this is that since we share this genetic code, we can actually, and a lot of scientists do this, can compare genetic sequences. So we can compare nucleotide sequences, as we've learned in the previous chapter, to look at differences between individuals or how individuals are related to one another, but we can also use them to create phylogenetic trees or relationships among species. So now we're going to move on to the 3 different types of RNA, and we've covered these a little bit previously. So we have our messenger RNA, so its job is to transport. Our ribosomal RNA that provides a surface or space for translation, and our transfer RNA that is going to move, as you can see here, our amino acids from the cytoplasm to its corresponding place along the mRNA transcript. And then I also mentioned there are other classes of RNAs, and their job is to help control the expression of genes. So here's a nice list of the different types of RNA and what their functions are. So our transfer RNAs, you can see, are the smallest of the 3, and messenger RNAs can be the largest depending on the gene sequence that needs to be ultimately translated. So our mRNA transports our genetic code, so it encodes our amino acid sequence. Our ribosomal RNA associates with proteins, which provides a place or location for translation to actually occur within the nucleus. And then our transfer RNA, its job is to transport the amino acids that are encoded by our codons. So we've talked a little bit already about mRNA, our messenger RNA. So it is essentially carrying some complementary information that is is complementary to our DNA template, remember? So it's complementary to that, or it's going to be the same as our DNA coding strand. Okay, so it carries this complementary information to the template that's going to specify the protein that needs to be synthesized. It carries that information, as you can see in this image, from our nucleus to the cytoplasm, which is where the machinery is for translation. So we've already discussed this. Point 2, 3 mRNA bases in a row form a codon. So even though we have the same bases in DNA, when we refer to a codon, we're specifically talking about mRNA and the transcript of it. And most mRNAs are anywhere between 500 to 4,500 bases long. So here we have our ribosomal RNA. So these are medium length, 100 to 300 nucleotides long. They associate with proteins to form the organelles called ribosomes. And our ribosomes are made up of 2 different subunits. So we have a large subunit and a little small subunit that we'll see later. Okay, so there are 2 subunits, and that come together during the second process of protein synthesis, so during translation. And these things actually provide the support. Sometimes I like to say they read the strand of mRNA, but they provide a location for which our strand of mRNA can go through and allow for our amino acids to be moved in via our tRNAs. Some other RNAs are catalysts. So we have ribozymes, which are enzymes. And there are others that actually help to align the ribosomes with our mRNA transcripts. Here's a nicer picture of a ribosome than what I just drew. So we've got a large and a small subunit that actually come together, and we'll see exactly when this happens, but during translation, these 2 subunits come together to provide the surface upon which the mRNA transcript is actually going to be read and paired up with corresponding amino acids. So you can see the larger subunit is the bigger of the 2, hence the name. So over 5,000 bases, and it's made up of about 49 proteins, because remember, it's not just RNA, it's also proteins. And the smaller subunit, a little under 2,033 proteins. Finally, we have our tRNA, our transfer RNA. And the job of the transfer RNA is to bring in the specific amino acid encoded by our mRNA codon. So as we can see here, tRNA is here. And it's got this thing on the bottom called an anticodon, which is going to match up and pair complementarily, and an amino acid. So these are small, so the actual nucleotide part of it is what goes around the outside of this big structure. So it's only 75 to 80 nucleotides long. It's described as this cloverleaf shape. In 3D, it sort of looks like an inverted L. So there are 2 ends, like I just mentioned. So it's got one end where it's associated with 3 bases. And these 3 bases, because they're complementary, following complementary base pairing rules to our mRNA transcript, we call these an anti-codon. Okay, so if the mRNA transcript has an A and a G and a C, that's my codon. The anticodon on my tRNA would be a U, a C, and a G. So this always follows our complementary base pairing rules, right? And this is on my tRNA. That looks terrible. And the opposite end of our tRNA, so on one end it has the anticodon, on the other end, this picture is nicer up here. We see that's where it carries the amino acid. So this particular tRNA with this particular anticodon is always going to bind on the other end with this particular amino acid. So we've got two pieces. So this just shows you a better image, zoomed in, of what each tRNA molecule looks like. So you can see this cloverleaf structure, and this is our kind of 3D visual. You can see how this cloverleaf is built up of, RNA, because it's got all of the bases here along the outside. And then we have two regions corresponding to our tRNA. So we have on our second loop this little ring, and that's where our anticodon is located. So in this scenario, our anticodon is AAG, and the other end, our 3' end here, so note RNA also has directionality. This is where the amino acid actually binds. So whatever AAG encodes, and if I look at my genetic code, because I don't know that offhand. And of course I can't find it. Okay, so AAG encodes the amino acid lysine. So it's always going to bond on this end quite strongly to lysine. So here down at the bottom, it just shows you what our mRNA transcript looks like, how we have these anticodons on our tRNA molecules that are complementary, right? And how those anticodons are associated with very specific amino acids. So that's why we say that our codons encode specific amino acids, because they're only going to bind with these tRNAs that carry complementary anticodons. So we know that these genetic instructions are carried within our DNA, and in order for phenotypes to be produced, those genes have to be expressed. The proteins need to be synthesized. And if this were happening to all of our genes at the same time, would not be a good thing. So we need to find a way that this all can be managed. Or that, for example, we've discussed how different cell types, even though they have the same genes, they actually express different subsets of them. So the way for this to be managed are by proteins that are called transcription factors. So these transcription factors come together, they interact, and they form the structure or apparatus that binds to the DNA at certain locations. And because they do that, that's what helps initiate transcription at specific sites when our cells specifically need it. So as I've mentioned, they're proteins, they interact with, and they also form this apparatus to bind at DNA. So we know our cells have the same DNA, they can carry out specialized functions, and they have different forms because they express different subsets of genes. And this is all due to these transcription factors. And that's because they bind at certain locations or specific sites on the chromosomes in order to signal, hey, this is where we're going to start protein synthesis. So how this works is they actually respond to signals from outside of the cell. And those signals can either be hormones or growth factors that are actually bathing the cell. And they form a little pocket for RNA polymerase to start binding and doing what it needs to do. So these signals essentially are letting the cell know, hey, something needs to happen. Our transcriptor factors come in. So these transcription factors provide a nice link between our genome and the environment. And mutations in these transcription factors can actually cause a wide range of effects. So now we're finally to the detailed steps of transcription. So we have 3 different steps. We have initiation, elongation, termination. So in initiation, it's essentially how do we know that everything needs to start? So we've got these transcription factors, which we've just discussed. And we also need RNA polymerase. So we've got the idea is that these factors and RNA polymerase need a clue. They need to know where to bind to the DNA to actually start transcribing. So both of these things are attracted to a region called the promoter. So the promoter is a special sequence, and because right now we're dealing with transcription, so we're making an mRNA transcript from our DNA template. So there's a specific sequence on our DNA template called a promoter. And this promoter essentially signals, hey, this is the start of a gene. So it's attracting the transcription factors and RNA polymerase to this region. So we've got, remember transcription factors, we also have proteins that come in. So the first one to bind is called this TATA binding protein. And it's attracted to a box. And once that first transcription factor binds, other ones come in. So that's why I'm listing here just transcription factors are attracted. So they go first, our transcription factors. Then our RNA polymerase comes in. So all of this, these transcription factors and proteins and RNA polymerase forms this big thing called a complex. And all of this is attracted to the promoter, so it's all binding at the start of the gene. Okay, so here I've got all 3 steps listed out. Right now we're focusing on initiation. So our RNA polymerase, the transcription factors and proteins are attaching to the promoter. So they form this big complex around the region that indicates, hey, this is the start of the gene. That happens first. Once everything is attached, the RNA polymerase is the enzyme, and it starts to unwind the helix. So it does so, and only 10 or 20 bases at a time. It's unwinding the DNA double helix, and it actually begins to start adding nucleotides. So our promoter is that special sequence telling us, hey, this is the start of the gene, and so it tells us which strand of the DNA is actually going to serve as our template, because it's telling us which of the 2 strands is going to be transcribed. So this image just shows you a lot of detail of what's going on with an initiation. So in eukaryotes, so like us, we've got this thing called a TATA box. And so I mentioned that the promoter is this specific sequence along the DNA that signals the start of a gene. So in this case, it's not just this box, but there's a whole string of nucleotides here. And all of them together form this promoter. But what we do see within the promoter is this special thing, special region called a TATA box. And that TATA box attracts one of these proteins that we need called a TATA binding protein. And the importance of that is this protein is attracted to the sequence, right? And attaches here. And once that attaches, it attracts our transcription factor. Okay. And not just one, but others and other proteins. But our TATA protein comes in first. It's our binding protein. Then other transcription factors come in, and all of that together, the proteins, transcription factors, those serve to attract our RNA polymerase. So once our RNA polymerase comes in and forms a complex with all the other transcription factors and proteins, that's when it can begin to unwind the DNA. So our next step is elongation. So we want to make this transcript long. So RNA polymerase moves along the DNA, our template strand, unwinding the helix a few nucleotides at a time, and adding RNA nucleotides that are free-floating within the nucleus according to our complementary base pairing rules. And it does this in the same direction that replication occurs. So we always have directionality with our DNA. So it does this in the 5' to 3' direction. So it's moving along, if it's building in the 5' to 3' direction, it's moving along the template that's oriented in a 3' to 5' direction. This is my polymerase, right? So it always builds 5' to 3'. And our final step is simply called termination. And this occurs once RNA polymerase encounters what's called a terminator sequence. So just like our promoter signaled for the start, our terminator sequence signals the end of the gene. And once polymerase reaches that, it detaches from the DNA. The transcript of mRNA can peel off, and our double helix can reform. So this schematic shows you all 3 steps: initiation, elongation, and termination, and how this process works. So remember, initiation, the big key is this promoter, right? So this is that region that signals, hey, this is where we want our TATA binding protein, transcription factors, and our RNA polymerase to all come together as a complex, right? And once that happens, RNA polymerase starts to unzip the helix, as you can see here, and starts to pair up bases complementary to the template that's exposed. And we've started. Elongation is just simply building on that process. So RNA polymerase keeps on chugging along in this 5' to 3' direction, unzipping, complementarily pairing the bases that are exposed. Once it reaches this terminator sequence, this special region that signals for an end of a gene, it's released. So if our RNA polymerase is removed, the whole thing's going to stop. So as that leaves, the transcript, all these bonds break so that it can leave, and then our DNA helix reforms. So this process of transcription is extremely efficient. So this just shows you that several different mRNA transcripts can actually be transcribed from the same template at the same time, because they can all come in and be attracted to different promoters and be doing their own thing, because remember, they're transient. So they can be copied at the same time. And so what this means is that many identical copies of the RNA can be transcribed. So if we need lots of the protein product, so they can be done so simultaneously. And this actually often occurs because mRNA, as we've discussed, is so transient and short-lived that half of it actually is degraded about every 10 minutes or so within a cell. So for gene expression to be constant, or if we need those proteins, we need lots of copies of our mRNA. So interestingly, I just want to point out that different genes on the same chromosome can also be transcribed from different strands of the double helix. So all of this, lots of different steps, lots of different genes, all of this can be going on at the same time within the nucleus. In eukaryotes, we now need this single strand of mRNA, our mRNA transcript, to leave the nucleus. It has to go to the cytoplasm because that's where the machinery is for the protein synthesis to actually take place. So there are several steps that then happen to this mRNA transcript before it becomes mature. So as it just leaves and peels off, this transcript peels off, once the polymerase reaches the termination sequence, several things happen for it to become mature. So a methylated cap is always added to the 5' end. And this cap is important because essentially it serves as a recognition site for protein synthesis to actually start. So it consists of a series of guanines, so lots of G's, that attract an enzyme that add methyl groups. So that's our methyl cap. At the 5' end, we add about 200 adenines. And that's called a poly-A tail. So lots of repeated A's, about 200 of them. The 3' end, it's our poly-A tail. And that component is necessary for protein synthesis to begin because it helps stabilize the mRNA so that it stays intact longer. So we need these two pieces. We need this cap, the 5' end. We also need this poly-A tail at the 3' end. So during this processing step, this is when we can take this mRNA transcript and alter it if we need to. And this is called splicing. So our introns are regions that we're actually going to remove. So they're little sections that kind of intervening bases that we need to pull out. And the remaining parts that are left over after that, we're going to splice that together. And those are called the exons. So introns are removed, so little segments that are between. Exons then are what are left. That's what we're going to kind of stitch or splice together. That is going to be what we translate into our amino acid sequence or proteins. So an interesting fact is to note that the introns, so the things that we're actually removing, can actually outnumber and even outsize, be larger regions than the exons are. And depending on which things we decide to remove, we can make different protein products. So at that point, the mRNA is proofread and everything's all mature at this point because we have the cap, we have the tail, we've got all of the introns removed, and at this point, the mRNA mature transcript can finally be sent from the nucleus to the cytoplasm. So I've mentioned this on the previous slide. So the splicing is the way that we remove the pieces we don't want. And alternate splicing just refers to a mechanism in which we can combine those exons, so the things that we're going to keep in, of a gene in different ways. And so this allows for different cell types to actually create different versions of the same protein. Right? So if we're expressing the same gene in two different cells, but we remove different parts of the sequence, so we end up with different exons that are there, we actually can create different versions of that same protein. So they can look a little bit different, and when they look a little bit different, they can function a little bit differently in the different types of tissues. So just a way, before I forget to remember, the exons are the things we keep in. Remember, the exome is the part of the genome that is actually expressed as the phenotype. So that's how this X kind of goes together. So here we have a schematic showing all of these different components I just discussed with the processing. So here we have our DNA template strand, and it's just showing that our template strand, the series of nucleotides along that template is made up of exons and introns, things we want to keep and get rid of. And we're going to transcribe all of it, all of the information into our mRNA. So this is our pre-mRNA, because you can see that it consists of everything that's complementary to what we started with. So the next step is to modify that, make it mature. So the way we're going to modify it, on the 5' end, we're going to add that methyl cap. On the 3' end, lots of adenine, so our poly A tail. And now we can finally splice. So we're going to remove, in this case, these 2 introns. We're going to completely take them out, stitch or splice the remaining 3 exons together. So that is the only region of the original DNA that we're going to maintain and pass on to help synthesize proteins. So that mature transcript is what we're going to send out into the cytoplasm. And this is where, if you alternatively splice it, just a note, say we wanted to remove exon B for one cell type, you could, and just stitch the last 2 together.